Today I'm going to talk about plasminogen, which is the unsung workhorse for fibrinolysis. And then I'm going to share some of the fundamental basic as well as some of the new developments we have in the laboratory. Before I dive in, I would like to thank our funding bodies, which is the Australian Health and Medical Research Council and Australia's Economic Accelerator. I would also like to thank the Australian synchrotrons for our access of bin time, and also members in the laboratories, whether it's past or present, who has contributed to this work. So perhaps you want to ask, why the basics? Because James and I, we started in this field as structural biologists, and inevitably we have been exploring into the intricate interplays between the structure and function and looking at its role in fibrinolysis, tissue remodeling, and then later on in bacterial infections. But you really can't look at plasminogen without putting it, and structure and function in particular, looking at its activations. And I know it's a very complex system, but just to dumb it down, plasminogen has to be immobilized on the surface, and then it got to be colocalized with either the tissue plasminogen activator or urokinase plasminogen activator before it can be activated. So once it's done its job, when it comes off the surface, then it will be immediately removed by alpha-2 antiplasmin. Going back to the structures, it is a 7-domain protein. At the N-terminus is the PAN domain, and upon its removal, it generates the lyse plasminogen, which is followed by 5 homologous Kringle domains, and it contains the lysine binding sites, allowing it to attach to the surfaces. All of them has got the lysine binding site except for the one in the middle. So the C-terminus is the functional domain, which is the serine protease domain. Its activation involves a proteolytic cleavage between the Kringle 5 and the serine protease domain at the activation loop, and these two chains remain stick together. In order for it to stop grabbing onto every single lysine residues in the circulation, it adopts a close conformation. So how does it work? Through the structural studies, we have identified that the Kringle 1, which is a stronger lysine binding site, is open. It allows it to mediate its docking to the target. And the remainder lysine binding sites are involved in interdomain interactions, and there are 5 of them. 3 of those was mediated by lysine binding sites, such as the Kringle 2 binds to the serine protease domain, Kringle 4 and 5 bind to the PAN domain. So without the PAN domain, plasminogen in its lyse form is open, and it shouldn't be found in the circulation. The remainder 2 was mediated by chloride ions, which reinforced the interdomain interactions between the Kringle 2 and the SB domain, as well as Kringle 4 and the PAN domain. As you notice, that the Kringle 5 binding to the PAN domain probably was the weakest, and it involved only one disulfide bridge, and therefore is the Achilles heel we expected. It mediates, the first step, it is involved in the transition from the closed to the open conformation. So if you look at plasminogen isolated from the plasma, it's far from homogeneous. Recently, we published a structure in collaboration with Albert Heck from Utrecht University. We set out to annotate and quantify all these subtypes, and we have identified a total of 14 of them. So are you ready for it? I'm going to tell you what they are. There are 3 glycosylation sites and 1 phosphorylation site. If we start with the Olin glycosylation T346, all plasminogen from plasma is Olin glycosylated at this site, and it either exists in 3 sugars or 4 sugars. And each of them can have an ending glycosylation at residue N2A9 that separate them into glycoform 1 and glycoform 2. Now, each and every single one of them can also have an additional Olin glycosylation at S249 or phosphorylated at residue S339. So at the top with the ending glycosylation give us the type 1, at the bottom without the ending glycosylation give us the type 2. Now one of the Olin glycosylation at T346 with the 4 sugars, when it's doubly Olin glycosylated, is underrepresented in the population. We have isolated, and therefore we have got a total of 6 type 1 and 8 type 2 altogether. So we have studied 6 individuals, and it tells us categorically that the type 2 plasminogen probably is 3 times more abundant than that of the type 1. A lot of the plasminogens are phosphorylated, which is twice as much as the unphosphorylated population. And lastly, only 20% of the native forms is doubly Olin glycosylated. So you want to ask what it is that PDM affects its function or its activation. So the first one is the T346 Olin glycosylation, which is found in all plasminogen, which is found right at the top of the activation loop in a place called the K34 linker. This Olin glycosylation actually locks this loop in place and prevents activator accessing the activation site, and therefore, hypothetically, we cannot activate, especially by TPA, the closed plasminogen. Having said that, it doesn't apply to streptokinase, which is a bacterial from Streptococcus pyogenes. It can wrap around the catalytic domain and activate plasminogen, but if you look at the rate of activation in the closed form, he's the old Adam over there who did most of this work, we can easily separate glycoform N2, and in the presence of streptokinase, in the presence of increasing concentration of the substrate, you can easily see the rate of type 1 activation is twice as fast as the type 2. So going back to the phosphorylation, although plasma protein phosphorylation is not common, but it is without precedent, we have seen phosphorylation in fibrinogen, factor V, and complement C3. Now, categorically speaking, they are responsible for signaling, localization, and receptor binding. We have spent a lot of time trying to work out what is this phosphorylation do to plasminogen, but we have not been able to put our finger down what it does. Having said that, we did identify the kinase which is involved in the phosphorylation. This is the Golgi kinase. It phosphorylates secreted proteins. And we identify it by the motif, which we carefully mapped onto plasminogen, which agrees well with the motif it recognized. What do we know about FEM20? Well, from our point of view, I sort of realized that its mutation has got to do with Raine syndrome, which leads to bone and dental mineralization deficiency. So, we also learned something new in this study, that the lyse plasminogen was generated in a 2-step process. First of all, it was cleaved after R67 and then leaving a beta strand dangling behind for the last cleavage on the canonical site after K77. So, although this 2-step processing has never been reported, but when you look at it on the structure sense, it makes sense. First of all, the R67 is on the surface. When it's removed, then it exposed the K77 residues. So taking it all together, we have identified 14 plasminogen proteoforms. The O-link glycosylation and residue T46 protects the closed form from activation, and type 1 plasminogen activates twice as quickly as type 2. We also reported that 70% of the plasminogen in the plasma is phosphorylated, and the cleavage of the PAM domain is a two-step process. So now we all know everything about plasminogen. Let's turn the table around and have a look at how it responds to ligands. In order to do that, we use small X-ray scattering. And here we measure the radius of gyration, the size and the shape of the molecule, and the maximal dimension in the solution. Now you can easily see that the closed form in red diffracts very differently to the open form in blue. So from this study, we have derived that the closed plasminogen has got a radius gyration of 32 angstrom with a maximum dimension of 100 angstrom, whereas that of the open form is with the radius of gyration of 49 and also a maximum dimension of 170. Now, how do we look at its response to the ligands? We have set up a high throughput assay. Now, if you focus on this one, in this closed form in bright red, if we increase a particular ligand concentration, it gradually turns into the dark red, which is the open form. Now, if we calculate each and every single one of the curve in terms of the percentage of open plasminogen, we can plot it against the ligand concentration, and from there we can derive the K to open value, which is the concentration of a particular ligand, which gives you 50% of the plasminogen assume an open conformation. Now here's a summary of a lot of the work that we have looked at various ligands, including on the top of the list, which is the VAC75 from bacteria, and also the last 50 amino acids from alpha-2 antiplasmin, with the VAC75 being the most potent ligand and lysine at the bottom being the least potent ligand. The differences between one to another is more than 1,000-fold. Now, does it mean that it can be translated to plasminogen activation? Using the same principle, we can also study the K activation. And from here, although the numbers are not the same, but you can see the change in magnification is very similar. So we're quite confident to conclude that this conformational change can be translated to its rate of activation. Now, not only it responds differently to different ligands, we've also observed that the open plasminogen assumes different conformations, and there are 12 of them being modeled. So we probably can speculate this different conformation allows it to bind to different receptors. Perhaps it's too bold to assume that those is a result of post-translational modification, which I'm not there to say it in here. So, different plasminogen receptors. We know that plasminogen receptors on the cell surface are kind of a mess because we can only define it as ubiquitously expressed and structurally diverse group of proteins. And there are a total of 14 of them has been described so far. Some of them are peripheral membrane proteins and the others are integral membrane proteins. Now, amongst those, RKT is the only professional plasminogen receptors been described so far. Not only they're very different cells when plasminogen bind to it has got different response. When it binds to injured cells, it produces pro-inflammatory chemokines to recruit immune cells to the injury site. Whereas if it binds to the immune cells, it can trigger the production of anti-inflammatory cytokines to promote wound healing. Now, The cell biologists know everything about how plasminogen binds to the receptors and their responses, whereas the structural biologists are lagging far behind. All we know is that it binds to the surface lysine and has got a very low affinity, which becomes a bottleneck for us to actually do any structural study to understand their molecular interactions. We can only look at the domains interactions. In this instance, we use the last 48 amino acid of RKT and SBR to measure the affinity. With the full-length plasminogen, you can see that the affinity is around low micromolar, as I mentioned before. So if we look at which of the domains involved, we can knock out every single lysine binding site in the full-length background and look at their binding. So if you knock out Kringle 1, it doesn't bind at all, which means that it absolutely needs Kringle 1 for the binding. Locking out Kringle 2 and 5, it doesn't really affect its binding at all, but to our surprise, if you knock out Kringle 4 lysine binding site, the KD has increased substantially. We can verify that Kringle 1 and 4 actually are involved in the binding of plasminogen by using the individual Kringle domains to repeat the experiment. As you can see that only Kringle 1 and Kringle 4, just those two, can bind to the peptide, but none of the other ones we have tested actually show any binding at all. Just to summarize, we've shown you that the kinetic of plasminogen conformation change is ligand dependent, and we think that can be directly translated to its rate of activation. We also showed you that the open plasminogen can adopt multiple conformation, which may allow it to bind to different receptors. The RKT binds to the lysine binding site located in Kringle 1 and 4. So we have to admit at this point that the mystery is exactly how plasminogen binds to its host receptor at the molecular level remains to be unraveled. I'm going to switch gear in here just to talk about, beyond the structural studies, what have we done? So we know that although we keep talking about the micromolar affinity, we can't do the structural studies. Bacteria hijacked plasminogen for invasion. In order to do that, they developed their very own very high affinity receptors. Now, just save an example in here. This plasminogen streptococcal M protein has got an affinity of 2.7 nanomolar, which is way higher than what we have seen previously. We have also shown that plasminogen binds to the spore of Clostridioides difficile to show that we use Western. E1 and E2 are two human isolates. And you can clearly see the plasminogen bind to it, and along with a confocal image where we use fluorescently labeled plasminogen. Using SBR, we can measure the affinity. Immobilizing the spore on the chip, you can see that it binds exclusively to human plasminogen, but not that of the mouse plasminogen at micromolar range. So this plasminogen bound, we have shown that it is activated to plasmin. You know that plasmin doesn't normally sit on the surface without doing anything. So we want to look at what does it do to the surface of the spore. Now here is a cross-section of the spore. On the outside is the exosporium, and further in is the spore coat, the outer membrane, and the cortex. The cortex layer shown as the white and dense color and the TEM. If we measure the exosporium with this square bracket in red, and the untreated spore, it measures around 74 nanometer, but the same has reduced after plasmin treatment to less than half of 35 nanometer. Does it promote spore germination? Well, we can measure spore germination by looking at the turbidity. Using this one as a reference, normally the reduction of germination or reduction of turbidization as this. Now, in the presence of plasmin, it reduced further, suggesting this thinning of the spore coat actually promote its germination. So this is the working model that upon infection, the C. diff produce toxin A and toxin B, which sort of cause damage to the epithelium. It allows plasminogen to accumulate at the infection site, which binds to the spore and promotes germination, dissemination, and also tissue damages. So we want to, and while we're trying to work out what is the receptor involved in plasminogen binding, we also want to entertain ourselves to see whether we can actually inhibit plasminogen activation, using an A here, or plasmin activity with an I over here to moderate the infection. We have two SCFV, which are specifically targeting the activation. One of the one binds to the activation loop over here. It stopped the activator cleaving the activation site. The second one binds to the catalytic triad, in particular the aspartate residues. So we use those two SCFV. We infuse it into the animal an hour before and 7 hours after the infection at 40 mg per kg. And looking at the observation at 24 hours and the viability at 48 hours. So here's the results. We monitor the physiological appearances, the cage appearance, and the fecal consistency. To our delight, the SCFA over there has substantially improved the presentation in all the parameters we have measured. Disappointingly, the SCFI, on the other hand, is very comparable to the naive SCFV control. If anything else, it exacerbated the symptom. So at this point, we have shown that plasminogen binds to the C. diff spore. Upon its binding, it remodels the spore surface and promotes germination and dissemination. We have shown also that stop activating plasminogen with the SCFVA has protected the animal and improved their survival rate. As why the SCFVI has lack of a performance at this point, we don't know. Our hypothesis is that it cannot be accumulated at the infection site. So the next study will be to have a look at its tissue distribution and have a look at whether or not we can administer it via a different route. So before I wrap up, I just want to quickly share about our latest approach in unlocking the plasmin activity in order to promote fibrinolysis with the aim of helping with thrombotic diseases. So traditionally, tissue plasminogen has been used. In here, we explore two alternatives. One is to use plasminogen, and the other one is targeting alpha-2 antiplasmin. In regards to plasminogen, we have created a range of different variants, and some of those actually can improve the half-life by more than 5 times. In regards to targeting alpha-2 antiplasmin, our lead compound actually have shown an EC50 range from 13 to 1.3, depending on the assays that we're using. Now, because Chris Barrett and also Lucy Coburn is going to tell you all the details of the experiment, so I'm not going to steal their thunders in here. To accompany with, to complement inhibiting the alpha-2 antiplasmin, we have developed a beta test, proof of concept, to measure the alpha-2 antiplasmin and the PAP in the plasma in 10 minutes. So Inisha is going to have a poster presented today, and we would really like to hear your feedback about the concept as well as whatever the translation value of that. So with that, I just want to thank all the members of the laboratory who have contributed. The one at the top over here, Adam Craig, Darshini, Jason Wu, and Paul Conroy. They have moved on to their new careers, and James is always the biggest supporter of this project. Our current member, Hui Li, Lian, Gordon, and Anusha. And here are our collaborators. In particular, I want to acknowledge Tom Cardock-Davis, who's always been very generous about providing the beam time. Chris Barrett, who's been helping us with looking at the plasminogen variants. Lucy Cochran, who's been looking at the alpha-2 antiplasmin inhibitor. Chris Langendorf is heavily involved in doing the mass spec study together with Albert Higg. Dina Lyra, who's provided the animal model for the studies. Rob and Christoph, who are also over here. So with that, thank you very much for your attention. I'm happy to take questions.